Welcome again to our session on science and public policy. Our second speaker today is Gideon Dunster. He's going to tell us about music in the brain, and Gideon's a biology major.
Well, thank you all for coming. As you said, I'm Gideon Dunster. I did my IS on looking at music, learning, and protein expression in the brain, and I was advised by Dr. Patrick Crittenden here in the biology department. So, what is the Mozart Effect? Well, the Mozart Effect was originally established in 1993 when a group of human researchers had a group of college students listen to one of Mozart's sonata for about 10 minutes, and then they tested them on a spatial IQ reasoning test. And what they found was that college students that were exposed to this Mozart music scored roughly 8 to 9 points higher on this IQ test than college students who had listened to a relaxation tape or had listened to nothing at all. And then in 1998, we got the exact same results translated in a rat model. So we had scientists exposing rats to Mozart's sonata and then testing them for spatial learning. And they found that rats exposed to this music performed better at their spatial tests than rats who didn't. And finally, in 2005, these results were translated once again into mice. So what we have is this idea that you can go ahead and take probably sub or regular intelligent people, maybe sub-intelligent people, and you go ahead and you add Mozart, one more please, all right, and what you get is, now this theory, when these results were published, this took off, and the public policy portion of my speech, I guess, would be tied to the fact that after these results came out, there were all kinds of industries set up for music exposure to young children. Anyone knows, I think it's Baby Beethoven. There was a law in South Carolina that all daycare centers had to play classical music when kids were at play. So there was a huge amount of industry generated from this one study. So why might this work? Well, in the rodent model, which is what I end up using, one idea is that music exposure serves as a form of environmental enrichment. And this simply means that say you've got 2 cages of mice, rats, whatever rodent you have. And in one cage you put stimulating objects or other rats. So what they get is they get increased cognitive stimulation from toys, from social interactions, from whatever you have, compared to this group of mice that might be housed alone or in what we call impoverished conditions. So essentially what researchers have established is that housing animals in an enriched environment causes increased cognitive awareness, better exploration in open field mazes. What you have is pretty much more active mice or rats. So it's possible that music may serve as a form of environmental enrichment. So how would this work? Well, anytime you talk about learning and memory, you focus in on the hippocampus. And this is generally accepted to be the major site of learning and memory in the brain. Now, as you talk to any neuroscientist, they will talk to you about associated cortices or other areas of the brain that do have memory and learning implications, but really the hippocampus is where it all comes back to. And so, specifically inside the hippocampus, I was looking at this molecule called brain-derived neurotrophic factor, which I'll abbreviate BDNF for the rest of this speech. And what it is it's a protein that is generated by nerve cells and released onto other nerve cells that has a whole host of consequences on those cells that receive it. And these aren't simple signals. This isn't an excitation or inhibition signal. This protein is associated with changing conformation of cells, the way that cells connect to one another. And by changing the way cells connect with one another, or synaptic plasticity, you can alter essentially the fundamental construction of portions of the brain. So perhaps BDNF, right, as a regulator of synaptic plasticity, is increasing this learning and memory. Well, this would all be great if everybody was able to replicate these experimental results. But we have a problem here. We have a debate. And when you look at the literature, since 1993, there has been a debate between these 2 sides of the argument. Groups that have been able to find or replicate the results of the Mozart Effect, and groups that have not. And so you have this field split pretty evenly into 2. People who believe that this is an actual biological phenomenon that can be tested and reproduced, and the fields that say, hey, we haven't been able to reproduce it. I think that what you have is an anomaly there, and it's not actually an established phenomenon. And so we have inconsistent results in both human and rodent models. And so this leads to a lot of questions about is the Mozart effect real? And if so, how is it being replicated? So we still don't have conclusive evidence one siding with one side or the other. So the goals of my study were first of all to replicate the Mozart effect. This type of study has never been done at Worcester before, and so I was planning on serving as sort of a pilot study for this type of experiment. And I wanted to just see, can I bring the result of the Mozart effect as shown by other people here to Worcester? And then if I could, I wanted to see if I couldn't compare spatial learning in a Morse water maze, which I'll explain soon, to protein expression in the brain. I want to see if I couldn't link changes in observable behavior with changes in protein expression in the brain. Finally, if I was able to do all of that, then I wanted to get down to investigate the music stimulus itself. You know, why Mozart? Why classical music? What is so important about this piece? I mean, for all that I know and as much research as I've done, the researchers chose it subjectively, just kind of on a whim. So, does the nature of the instrument possibly play a role? Could it, you know, what part of music is eliciting these responses? So what I did was I took 32 mice and I split them up into 4 groups of 8. One group got a control, they didn't listen to any music. One group listened to Mozart's sonata. And then I had 2 other groups that listened to Samuel Barber's Adagio for Strings or Agnus Dei. And the only reason I chose these was because they are roughly the same length as the sonata, and they each use a different instrument. So Mozart's sonata is played by 2 pianos, Adagio for Strings is played by all stringed instruments, and Agnus Dei is by voices. It's a vocal piece. And so it allowed me sort of an internal control of the music stimulus itself, which may help me, if I got positive results, to look at what part of music might be increasing learning and memory. So what I do is I house these mice in these cages, and you can see they have isolation chambers. This is a plexiglass cage, and there's 2 groups of 4 mice each, and in the back you see speakers. And I played this music for 12 hours a day for about 30 days during their active cycle. Okay. At the end of those 30 days, I tested them in what we call a Morse water maze. Now, for you non-behaviorists out there, the concept is relatively simple. What you do is you take a circular pool of water, and you put a platform, and then you opaque the water so that when mice are in it, they can't see the platform. And the goal is to have them find where the platform is without being able to see it, relying on these close cues on, the platform. I can see it's right there. We're trying to remove that and have them rely on distal cues around them to see if they can't find where the platform is. And once they get on the platform, they get removed from the water. So over 4 days, I trained the mice to find this platform. I kept putting them in, had them find, once they found the platform, I would take them out. On the 5th day, I did what's known as a probe trial. And I was sneaky. I took the platform out of the water maze. And the mice didn't know that. And what I did was I would record each mouse for 60 seconds and see where did they swim. So here I show you how I broke up the Morse water maze into 4 quadrants, and I looked at how much time the mice spent in each quadrant. And you would expect if they learned the position of the maze, they would spend more time in the quadrant that previously held the platform than any other. Once I was done with that, I did a tissue extraction and protein assay. So I extracted the hippocampus from each mouse, and then I did a total protein assay and a BDNF ELISA. And essentially this means that I decided to look at how much total protein do we have there, and then how much specific protein do we have there. That way I could report BDNF, as you see, as a function of the amount of BDNF per total protein in the brain. So what do I find? Well, here is the result of my training days. And the take-home message here is you see that the slope of the line is negative. At the beginning of training, it took the mice longer to find the platform than it did at the end of the training. So what we have here is evidence of learning. The mice are learning where the platform is. But interestingly, if we were to collapse these lines and look at them, look at the slopes, we would see that there is no group that is learning faster than any other group. So exposure to any forms of this music did not result in increased learning over the training days. Okay? So then I looked at my probe trial data, and I found similar results. So I looked at how much time they spent in each quadrant, and I found that, once again, no group spent more time in the target quadrant, the quadrant that used to hold the platform, than any other group. So once again, we see that exposure to any of these types of music did not show increased memory of where the platform was. So I finally looked at the protein results, hoping that maybe something would come out of them, but once again, we show that there is no difference in the protein expression in the brains of the mice either. So no group had more or less, significantly more or less BD and F than any other group. So this leaves me with these conclusions. I don't have any significant behavioral results. Exposure to music didn't lead to increases in learning or memory, nor do I have any molecular results, significant molecular results. Exposure to music did not result in increased BD and F concentrations in the hippocampus. So I wasn't even able to look at my other two questions. So based on these results, personally, I cannot accept the Mozart effect as a true biological phenomenon. So for future work, there is a lot that needs to be done, and a lot of it has to do with methods. Currently, there's no standard for how to perform the Mozart effect. Every study uses a wide range of animals, different types of animals, different species of animals. They use different forms of music. They use different times of music exposure, some for 30 days, some for 80 days. They use different behavioral tests. They use different molecular tests. It's just all over the place. And so we don't have an organized way of looking at this question. And in addition, perhaps we just need to start more simply. We need to break down this phenomenon into its parts to see if there really is something to study here. We need to look at possibly the components of music that might be eliciting these responses. We might need to look at transmission pathways in the brain or understand plasticity better before we can look at a larger picture altogether. So I just want to thank you for coming and thank Dr. Crittenden, my advisor, and Dr. Stabneser in the neuroscience department for serving as a pseudo-advisor for the behavioral aspects of this project. And if there's any questions, I'd love to hear from you.